Dr. Elizabeth Blackburn revolutionized science and medicine with her discovery of telomeres and telomerase, providing crucial information about chromosomal function, cell division, gene expression, and more. Dr. Blackburn was born in 1948 in Hobart, Australia, where she grew up around her parents and many other adult relatives who were all doctors, giving her an early introduction to medicine and science. This led her to fall in love with nature and animals to the point where she would pick up jellyfish at the beach and keep jars of tadpoles in the living room. In addition, she developed her own biological role model, Marie Curie. Later, she went to the University of Melbourne, where she earned her undergraduate honors degree and master's degree in biochemistry, and after to Cambridge University in England, where she got her PhD in molecular biology whilst working in the laboratory of molecular biology. She then did her postdoctoral research in Joe Gall's lab at Yale, and after that was rejected from many assistant professor positions. Ending up at the University of California, San Francisco in a research track position, she discovered and experienced gender discrimination in science, especially when she was later the only woman to be appointed to a chair position. As a person who loves challenging her brain, Dr. Blackburn has developed hobbies including piano and camping at a younger age, and even Sudoku later in life. Her dedication and determination to continue the research that started her career are particularly admirable. In one experiment, she found that if telomere levels were high and long in length, it meant that it was a cancer cell and promoted certain cancer-related characteristics such as excess tissue growth. Therefore, she developed an idea to use telomerase to attack the cancer, which is now in an early preclinical stage. As all my grandparents have battled from some sort of cancer, these mutations to telomerase bring me hope that there will soon be a better treatment and maybe even cure for cancer. Dr. Blackburn is in the field of molecular biology, specializing in telomere and telomerase biology. Her discoveries of both the chromosomal end cap and the enzyme that makes them have allowed for further research in cell aging, cancer, and gene therapy. It has also allowed for continued findings about the chromosomal end cap and chromosomes in general. In 2023, Dr. Blackburn co-wrote an observational study titled Predictors of Long-Term Neutralizing Antibody Titers Following COVID-19 Vaccination by 3 Vaccine Types, the BOOST study, published in the National Library of Medicine. This study investigated the durability of neutralizing antibody response of 3 COVID-19 vaccines and how factors like age, body mass index, or BMI, gender, and smoking affect these neutralizing antibodies. The 3 COVID vaccines include a single-dose viral vector vaccine, Johnson & Johnson, in addition to the 2-dose mRNA vaccines, Pfizer and Moderna. Key findings of this paper are found in the following figures. Figure 1, which compares the time after the vaccine series at 1 month and 6 months to the development of neutralizing antibodies, shows that for those who received Johnson & Johnson, the neutralizing antibodies increased over time, while for those who received Moderna or Pfizer, the neutralizing antibodies decreased over time. At 6 months, both the Moderna and Johnson & Johnson were superior to Pfizer, but the Moderna was superior to Johnson & Johnson. In Figure 2, they observe how age affects the neutralizing antibodies. The relationship between age and neutralizing antibodies was most negative for Johnson & Johnson, then Pfizer, and not significant for Moderna. This ranking of vulnerability to age mirrored the rank of neutralizing antibodies produced at 1 month in Figure 1, suggesting that vaccine potency may have protected against the effects of age. Furthermore, in Figure 3, they look at how BMI affects the neutralizing antibodies. BMI exhibited a significant relationship with neutralizing antibodies for Johnson & Johnson, but not for either of the other vaccines. Finally, in Figure 4, they saw how sex and smoking affected the neutralizing antibodies. In this, they discovered that there were higher levels of neutralizing antibodies in females in Part A and a higher level of neutralizing antibodies in non-smokers compared to smokers in Part B. Overall, the study concluded that across the 3 vaccines, they had similar durability of neutralizing antibodies by the 6-month test, after the Pfizer and Moderna mRNA vaccines produced more initial response that decreased over time, while Johnson & Johnson viral vector vaccine increased. They also found that older age was a strong predictor of neutralizing antibodies and that there was a negative relationship between BMI and neutralizing antibodies among Johnson & Johnson recipients. Dr. Blackburn's discovery of telomeres and telomerase earned her a Nobel Prize, allowing for many more discoveries in molecular biology. She has laid the groundwork for further investigation into manipulating telomeres and telomerase, and has also opened opportunities for the advancement in many research fields, especially cancer and aging. As she furthers her own research, she continues to study her own discoveries of telomeres and telomerase in her lab at the University of California, San Francisco. Dr. Blackburn's long journey in science, from the day she lived in Australia to the universities in the U.S., has shown her dedication, determination, and commitment, allowing her ideas and research to progress many other research fields and medicines. Thank you for listening.